Give me a second. Well, thank you everyone for coming for this talk, and thanks to Brian and Dusit for inviting me for this first talk for Sunbear expert team. In next 30 minutes or so, I'm going to talk about how leeches can help in developing a monitoring tool for sunbears. So this is something that we did as an exploration for last few years. Just to give some information about myself, my name is Sandeep Sharma. I work at German Center for Integrative Biodiversity Research here in Germany. I also co-chair a Bear Specialist Group's Asian Bear Monitoring Expert team. Um, so all of us know that we are going through an unprecedented loss of biodiversity extinction, and it's much higher than the background rate. So the picture that you see on your screen is from a paper that was published in 2015, and it talks about cumulative extinction of most of these vertebrate species, and
Sorry,
mammals are.
Sandeep, did you share something? I didn't see it.
Oh, you didn't see it?
Yeah, I just
Oh, sorry.
saw your face.
Okay, I'm so sorry. I think let's start again. I'll I'll basically skip the first few things. So let me, yeah.
Yep.
Can you see it now? Can you
Yeah,
see it now?
it's slideshow now. Yes.
Okay, so the first
Thank you.
slide I already spoke about it, but the second slide I'll go through it very quickly. So biodiversity extinction, this is what I was talking about. So mammals stopped the cumulative extinction rates, which was evaluated in this paper in 2015. This was done for all vertebrates. And as you can see that the extinction rate for mammals specifically was quite high compared to background rates. Now, why monitoring is important? Because if you want to reverse these extinctions, we devise certain techniques, we devise plans to reverse the extinction, and to monitor that progress, to monitor the effect of those efforts, we need to monitor and effectively monitor. So to assess the status, basically spatial distribution and trends of these species, to determine their demographic and anthropogenic factors that can influence these population variations in those species of concern. And it's essential for conservation science, because if we want to make any data driven decision making, and if we want to implement some proactive conservation actions, we need to monitor, and we need to monitor effectively with the methods that will give us more accurate results that are comparable across sites. And we also need evidence-based conservation efforts for this kind of adaptive management. So if things are not going according to what we wish for, then we will have to do some interventions. And for certain species where sustainable use is still permitted, again, monitoring is really important, because without that, you can't really set the quota for harvesting. It's not the case for bears, though, but I anyway wanted to mention about it. And then eventually spatial prioritization of habitats, because we don't have enough habitats for most of these species. For bear species, we don't have enough habitats in Asia. We need to designate certain protected areas and take some stringent measurement for their conservation. So even for that, monitoring is really important. Now, this paper that was published in Global Ecology and Conservation in 2022 by Garshalis et al., um, what you see on your screen is range map of these 5 Asian bear species, 5 endemic Asian bear species, except brown bear, which also occurs in Europe and North America. So if we talk about giant panda, sloth bear, sun bear, and Asiatic black bear, you can see those colors on your screen. They are mostly found in South Asia and Southeast Asia. The habitat there, you can see, is quite fragmented. And it's just so because of all of these anthropogenic factors that are causing habitat loss. Um, so I'm basically focusing on this specific area for my next slide. And what I'm going to show you over here is night light data for that part of the world. And as you can see here, that South Asia, mostly dominated by India and other countries, neighboring countries, Bangladesh, Pakistan, Nepal, Bhutan, and South Asia, they are heavily lit. and it's an indication that there are a lot of people there. So just this part of the world has almost 1/3 of global population living in just these confines of South Asia and Southeast Asia. And of course, those people, they have their requirements for infrastructural development, and that's why we have these severe cases of habitat loss for most of the species that reside there. Now, coming back to Asian bear species, there are a lot of challenges in monitoring those species. Most of them, they occur at low densities. Many of them have low detectability issues, very large home ranges. So, some of these conventional methods don't work in their case. And last issue is logistical issue or resource constraints. We don't have enough capacity to monitor these bear species. We don't have enough funding resources in Asia to monitor these bear species. So I just did one very basic analysis yesterday to find out to see the comparison. I did a Google Scholar search with the terms American black bear population monitoring, brown bear population monitoring, brown bear specifically for North America and Europe. And in this figure that is in front of you on your screen, you can see that American black bear and brown bear is very high as compared to all Asian bear species, all endemic Asian bear species. So we really need to step up in our efforts to monitor these Asian bears. Now, coming to sun bears, which is the topic of today's talk, we know that it's vulnerable. We know that its habitat, mainly forest habitat where it resides, is under a lot of anthropogenic pressure. And one thing that I would like you to notice here is on your screen is number of mature individuals. We don't have that information. We know that the population is decreasing based on expert evaluation, but we don't even know how many sun bears actually exist in the wild. And without that information, whatever we are doing, it's like that Chinese proverb where we say that flying a kite without a string. We don't have that string about population numbers of those individuals. And then there are a lot of threats which are increasing day by day. So of course, forest fragmentation, forest loss, mainly in Sumatra and Borneo and mainland Malaysia because of oil palm plantation and other infrastructure development. So they are causing severe threat to these populations. And then poaching of adult bears as well as poaching of juveniles for pet trade is another big issue that is causing threats to their populations, wild population. And then we don't have information. So, we know that sun bear populations are declining at an alarming rate. And there was about 9 to 22% decline within a period of about 14 years. And the same study projected a decline of over 50% between 2000 and 2030. So these are the projections. We know that situation is it's not looking positive. But we also have paucity of systematic and range-wide population monitoring effort for these species. We don't know the numbers, as I've said. And in fact, the systematic ecological research on this species barely started 2 decades ago. So we know nothing about these species, in my opinion. Taking you back to Dave's paper in Global Ecology and Conservation, where he and the team synthesized about 102 published studies that have generated information on bear presence, their distribution, occupancy, and some sort of population estimate or population trend. So as you can see here in green, we have very less information about population estimates. Most of these studies have focused on determining presence, determining distribution of these bears using occupancy framework to find out presence absence of these bears and some information about population trends. But then moving forward, what kind of methods these studies have used? So the figure that is on your left about percentage of studies that have employed different methods, you can see that these are dominated by studies that have employed interviews and science surveys. Some of these methods where you can identify individuals using camera traps or genetics are very few. And then when we break it down for different species, for sun bears, you can see that those methods are even fewer. So camera traps with ID, There's just one study that was done by Dusit in 2012, published in 2012. And while I was talking to Dusit in 2019 in Taiwan, he explained that this study was logistically very difficult because he was trying to identify bears, Asiatic black bears and sun bears, using their chest mark, the blaze on their chest. And for that, the bears have to stand up to get that picture. And then he had to bait those sites and put 3 camera traps. So logistically, it was extremely challenging, and it creates problems. It creates issue when you want to replicate these kind of studies in other countries within the bear range. I'll give some information about this global status review and conservation action plan for sun bears that was published in 2019, which emphasized the need for devising and employing reliable monitoring methods for sun bears. And then the objective 11 of this action plan also emphasized on developing population monitoring methods that can reliably detect these changes of sun bear population. And then developing and providing some of these effective monitoring protocols. And I'm emphasizing on effective because we do have methods, we have camera traps, we have DNA-based methods, but in tropical conditions, they don't work. I have worked on sloth bears that occurs in dry forest in India, Nepal, and Sri Lanka. So central Indian forests where we did our study, you could use scats and get DNA information from that. But doing the same thing in tropical condition is very, very difficult. So we need methods that would work in tropical habitat. And then building capacity for population monitoring, which is lacking, severely lacking in Asia. So this entire idea about connecting leeches with sun bears, it emerged in this workshop that was held in Taiwan in 2019. And I would like to thank Mishu and Dave for organizing this workshop, where a few of us met for a few days, and we discussed multiple things about how we can monitor Asian bears, what methods are there, if they are effective or not. And then this Asian bear monitoring expert team was also a product of that workshop. But for me personally, that workshop was very important and interesting because I met some of the collaborators where this entire idea generated. We knew that iDNA is not new, but to use it for tropical bears was something that was never tried earlier. Now, why do you see a brown bear, Himalayan brown bear, on your screen? Because when I participated in that workshop, we had just concluded a study. We had just finished fieldwork of that study, not concluded, fieldwork of a study, trial study on Himalayan brown bears. We also don't know about Himalayan brown bears. So we wanted to try out this method of high throughput sequencing of microsatellites. And I'll explain that method in a short while. We wanted to try out that method to see if we can identify bears, what's the accuracy of this identification, and if it works or not. And one of my collaborators, I think, is participating in today's webinar. So Dr. Vipan Rathore, Martha D. Barba, Trishna Datta, and I, we conducted this study in one of the protected areas in Himalaya in India. The habitat was really interesting because you can easily find scats. And here I was collecting scats. But to explain you about high throughput sequencing method here, unlike conventional sequencing, you do all the processes of DNA extraction and PCR and making libraries, but instead of calling the alleles in fragment analysis, you are actually sequencing them. And without going into too much of technical details, I'll just tell you what's the benefit of doing that. Because once you sequence that, you can sequence short markers. And that's very important because in scats, in non-invasive samples, scats, hair, saliva, DNA is highly degraded. So if you are targeting large markers, then you can't really amplify that. So short markers, it would result in higher amplification rates. Your genotyping errors are less because you're sequencing them. The allele identification is not it's non-ambiguous. You can standardize your allele calling. That means you can compare various different study if they are done on the same kind of technique. And then there are some other benefits, like you can detect hidden polymorphism, and it's quick and cost-effective way of large-scale analysis if you want to do it for larger areas. So very quickly, the results, we collected 74 fecal samples, and I won't go into details. We amplified 14 loci, but the point here is that the amplification success was quite high. It was almost 90%. So when I compared that with our previous work on sloth bear DNA study, our success rate was 50% because we were using conventional method. But whereas here the success rate was quite high. Now that was the point that created that connection with leeches, because here we had very high success rate now in the short fragments. And then I had seen this paper that was published earlier in 2012, where they did a pilot study of screening mammal biodiversity using DNA from leeches. It was one of the pioneering papers. And they found out that from leeches, you can identify certain species that occur in that habitat. So you can basically do mammal biodiversity monitoring of the area using leeches. And they also did this time constraint study where they collected leeches and they culled them at different time period. And they found out that you can detect the DNA, target DNA, even after 3 months. So, so that's where we started. We knew that our Asian bears, they occur in Southeast Asia, South Asia, mostly in tropical habitats. And then if you look at the distribution of these leeches, these hematophagous, blood-feeding terrestrial leeches, their ranges, they overlap. So why can't we use leeches as a tool to monitor these bear species that occur in these habitats? So not just the species identification, that's not novel, that's nothing new, it's been done. But going one step forward and identifying individuals, that was the challenge. So we started our study, we got some funding from IBA, and then the first study was done here in Europe, where we collected samples from captive bears, 5 different zoos. They provided us with their samples. We took help from European associations of zoo and aquaria. We got a blood scat and hair samples of 13 individuals, and we did high throughput sequencing of those individuals to find out if these loci that are developed on brown bears can work on sun bears or not. And they worked. So then the second step, we took this study to a field site for a controlled experiment. So we went to Bornean Sun Bear Conservation Center in Sabah, Borneo, and we took help of Wong, our colleague. And we also had a collaborator in Sunway University in Malaysia, Dr. Shamla Ratnaike. So we had one student, master student, who did all of this field work over there. And he took up this study as his master's thesis work, where we basically, as I said, controlled study. So when the bears were taken out for their regular medical examination, we released some leeches on the bear and let those leeches get the blood meal from those bears. By the way, these leeches were collected from the forest and they were monitored for a few days before we released them on the bears, because we wanted to make sure that the leeches are not sick or they're not, fed. So once we did that, we let those leeches feed on these bears for a few minutes. And then once they were fully engorged, then we started culling them, and we started culling them on typical interval. So here is our student, master student Terence, very meticulously collecting the data and rearing those leeches. I'll explain that experiment in a bit. And then we also collected dung beetles. So why these different ways, these different samplers? Because as I said, we considered this study as pilot study. We wanted to find out if leeches can identify species, if leeches can be used to identify individuals. And with dung beetles, because dung beetles were quite abundant in the region, if we can use dung beetles again to identify the species from the area. But along with that, we also did one very small experiment. I won't go into details of that work, but we also tried to find out if sun bears can be detected from their fresh scratch marks on the trees. So the result was, in short, was yes, you can, if the scratch marks on the trees are fresh, you can detect sun bears. But then going back to leeches and dung beetles, so with these two things, scratch marks and dung beetles, you can identify species ID and you can use that for occupancy. But with leeches, you can not only identify a species, but if you can identify individuals, you can use that in these spatial capture framework to obtain density estimates. So we we had leeches on 8 individual bears, 4 males and 4 females. And then there were several leeches that were released on these bears. So we started culling these leeches after predefined period. We were rearing them and culling them after 3 days, 7 days, 15 days, 30, 60, and 90 days. And some leeches even survived up to 120 days. So some leeches were culled exactly on the same day, and then we followed this pattern. For dung beetles, we did the same thing. We culled them up to 72 hours. And coming to some of the results. So these results, by the way, are still preliminary. We are still getting more data. But what you see on your screen is this qPCR results. So we use qPCR, and I would like to thank my colleague Angeliki, who's here, because she provided us with the primers that she had developed for sun bears and for Asiatic black bears. So we used her primers, sun bear primers, for this qPCR over here. Now on y-axis on this graph, you see CQ value, which is quantification cycle. So this quantification cycle would give you an idea. If it's lower, that means you have large quantity of DNA in that given sample. If it's lower, that means you can identify the species in very short frame of time from your sampler, which is leech in this case. On an x-axis, you have CQ 0, 3, 7, 15, 30, 60, and so on. So these are different time periods when these leeches were culled. So you can see here, for 0 up to almost 15 days, the uh, the margin of error was quite less, and the CQ value is 20. That means up to 15 days, there was ample amount of DNA in those leeches to detect the species. And moving on 30, 60th, 90 day, you see a lot of variation because the DNA quality probably starts degrading because the leech is also digesting those blood meal. Now, moving on to HTS, the high throughput sequencing genotyping result, the main question about identifying individuals. Again, we got very interesting results. So these results that you see on your screen are about those 8 individual bears for various number of days. So on axis, you see number of days from 0 up to 120 days. And on y-axis, you see quality index, which will give you an idea about how good that genotype is. So if you get full genotype, if you get the full identity of that individual, it's almost 1. But it would degrade if the quality index is going down, that means your genotyping is not accurate. Your genotyping has some errors. So you might get partial genotypes. Now here you see these different lines that are showing you that with the number of increasing days, the quality of genotype is decreasing, but they're still very good for certain period. So for that, we combined all of these results for different individuals because, of course, here you have variations that was introduced by or induced by different individuals. Now, summing up all of these results for 8 individuals, and here I would like you to pay attention to the positive, which is on the extreme right side. So the positive is actually blood sample from those individuals. And that you can consider as the gold standard. So anything below that means the genotype is not perfect. But anything above it or close to it, that means the genotyping is perfect. So you can see here that even up to 7 days after that blood meal, the genotype was as good as the blood sample that was taken directly from the individual sun bear. But even after 15 days, 30 days, the quality has gone down just by 20%. So you still get close to complete genotype. And you can still detect the sun bears individuals with some partial genotype up to 3 months, up to 90 days. Of course, with 120 days, the quality goes down drastically. So second phase that we started earlier this year, we had another grant from IBA, and we started this second phase where we wanted to do a quasi experiment. So now we are not, it's a semi-control experiment where we are collecting leeches from the enclosures of these bears at BSBCC, and we are also sampling within 5 kilometer radius of BSBCC. And this time, we also included other samplers, other iDNA samplers. So we already experimented with dung beetles and leeches, but this time we included mosquitoes because we recently did another study here in Germany linked with mosquitoes for mammal monitoring. And then these flies, you have carrion flies and several other fly species that feed on these dungs and on the blood of these species. So we had these 4 different kind of samplers. We sampled for 6 to 8 weeks. And this time we also sampled repeatedly within the enclosures and outside to see if we can identify the individuals, if we can identify the species. And if we can do that, then we can use this work in ACCR framework. So that work was completed, the field work, in fact, was completed just 2 weeks ago by one of our undergraduate student who took up this work as her undergraduate thesis. Her name is Sui. And now we are in the process of analyzing these samples. So what's the next steps? Our study was, as I said, very basic exploratory study where we wanted to answer a few very basic questions. But as next steps, if we want to convert this method as a replicable method, reliable and accurate method for monitoring sun bears, we need to replicate this at other location. We need to scale it up for larger areas. A field trial is essential. And then if we can combine this with other methods, because this method alone will give you certain amount of data. But if you can combine this with camera traps, if you can combine this with, if you can find scats, and if you can combine this with scats, you will get much precise results for density estimates. And our ultimate goal is to do population monitoring of sun bears in a given chosen landscape or protected area within the range of sun bears. And the ultimate goal of this work is capacity building in Asia and Southeast Asia. That's why we have these 3 students from Malaysia, because without local capacity building, we can't do much in these landscapes to conserve these species. So with that, I would like to thank my collaborators. First, I would like to thank these 3 amazing students. Terence, who took up this work for his master's thesis. He's just about to submit his thesis. He was supposed to submit his thesis on 30th of June, and that's why I scheduled this talk on 11th of July, but he's going to submit his thesis soon. And Shengyi, who did the dung beetle work, Sui, who did the phase 2 work, finished the phase 2 work recently. My collaborators, Shamala and Wong, who actually collaborated in the phase 1 work. And phase 2 work, our collaborators are Dusit, Trishna, and Union from Sunway University. And with that, I would also like to thank staff and veterinarians of Bornean Sun Bear Conservation Center, because this work would not have been possible without their excellent support. I would also like to thank Lydia Coulter and Jus Koch from Captive Bear Specialist Group and ESA's Taxon Advisory Group. All the zoos here in Europe that have provided us with sun bear samples. My colleague Marta De Barba and Tomasz from Slovenia, Divas Lab Slovenia, for providing us guidance about the analysis for high throughput sequencing. Thanks to Angeliki, who provided us information about these primers, because most of the time the researcher, carnivore researchers, whatever they discover, and if they don't publish, they keep it close to their heart. But Angeliki was very generous in sharing this information. We would also like to thank Sabah Biodiversity Council and Sabah Wildlife Department for providing us research permits, IBA to provide us the most crucial part of this work, funding. Without that, it would not have been possible. Sunway University provided assistance, financial assistance, and logistic support to the students and for our work, and eventually Bear Specialist Group for organizing that workshop in Taiwan, and excellent colleagues from Bear Specialist Groups who are a group who are always there to collaborate and help in whatever we want to do. So with that, thank you so much. If there are any questions, I would be happy to take those questions. Thank you.
Thank you, Sandeep. So I'll just open for question now.
Thank you, Sandeep. I'll jump in with a question. That was very interesting, all of it. Do you have an idea of how much money it would cost to scale this up to, let's say, the size of a protected area in Malaysia, for example?
It
Or
really depends. Sorry, I interrupted you.
yeah, specifically for monitoring, but yeah, you get the idea.
So it really depends on how big area that is. So if we divide that area into different sampling grids, and if we sample there for about 3 to 4 months, I would say again, I'm just doing some math about the numbers of samples and how much money we would need to analyze those samples. About, I would say, $50,000 for a medium-sized area. And then if you want to combine that with camera traps, because if you have data from other sources from camera traps, you can also do this integrated population models, which would increase your precision. So if we want to do that, then we need more money for camera traps and for monitoring those camera traps. I think logistics is one of the biggest challenges there.
Sandeep, when you say moderate size, what you mean by in terms of square kilometer, like 100 square kilometer or what?
Dusit, you're asking about the medium size, right?
Yeah, you mentioned the medium size.
I would
What you
say about
mean is like
300 square kilometer, but I don't have much idea about tropical forest conditions. So I think that's where we need some insights from experienced people like you.
Yeah,
And this
300
is
combining with
yeah
camera tab is quite well, I would say medium to large.
yeah no when I said medium, it's from my experience in India and here in Europe. So of course,
Okay.
300 probably is much bigger in case of tropical Asia.
Yeah, logistic. Yeah.
But
Yes,
I think
Sandeep, this is,
sorry, Dave, go ahead.
yeah, maybe you can stop sharing your screen just so we can
Mhm.
see people.
Is that
Thanks.
good? Yeah.
Yeah, that's great. I was gonna, first of all, compliment you on a wonderful talk, scientifically rigorous. And also, I think you could give a whole talk on what it takes to kind of collaborate to get to this stage. It was an amazing collaboration among many different people. I was just going to ask you a question about like the life history of a leech. How long do they stay on? How much do they feed? When do they fall off? How long do they live? And would this have been complicated if the leech had fed on other critters, other animals?
So I'll answer your last question first, Dave. First of all, thank you so much for compliments. Now, with your last question about, it was about, sorry, I forgot forgot it.
So it was about, feeding on another species or other animals. Would
Yes,
that have complicated this?
yes, thanks. So here, our approach was very targeted. That's why we use qPCR. You can use some markers that would give you information about the entire mammal community. But in that case, you would lose the signals of those species that are extremely rare. And I'll quote these two papers when we were thinking about this entire idea, and we discussed this idea there in the workshop also in Taiwan. And that time, those papers were already published. So there was one paper from Borneo, Drinkwater et al., and they found out in their entire study where they are explaining all of these different species that they found, in one sample, they found sun bear. And at the same point of time, there was another study from China where in a few samples, out of thousands of samples that they have analyzed, in few samples, they found Asiatic black bear. So that gave us the idea that, we have to have targeted approach. And now if we have targeted approach, it's like we are searching for the needle in the haystack. And that's what we did with this qPCR. So if you have targeted approach, if the leech has fed on other species a few weeks ago, and fed on sun bears recently, or even a few weeks ago, you can still detect that. Now, other questions. So, I myself am struggling with for the answers of those questions because, leech biology is something where at least I don't know much about. And again, we need to do some trial for that. So any one of you, if you know about leeches, please share the information. Or if you know of any leech biologists, please share the information, because we really want to devise this. I didn't get enough time to give information about the second phase, but in the second phase, we devised traps for flies, for mosquitoes, and of course for dung beetles. Dung beetles are easiest to trap. We get, within 6 weeks period, we got about 300 dung beetles. But mosquitoes were difficult. Flies were a bit easy than mosquitoes, but leeches, we don't have any traps. So if you know of any design or any innovative way of trapping leeches, then we can actually have a systematic survey for these iDNA samplers.
Yeah, I was just going to follow up. Another, the way that we normally survey bears is to try to, you can either do it as a random survey, or it can be a thing where you use bait to attract them, like for hair traps. And likewise, it sounds like you could do that with dung beetles, because you could attract a bear to a place where you would hope that they might defecate, and also attract dung beetles to the same site.
Yep.
I just don't know how you would do that with leeches to kind of cause the leech to fall off in a certain place or whatever to attract the leeches, but maybe you could.
Yeah, I mean, we are, that's what I said, we are still struggling with systematic sampling devices for leeches. Because for mosquitoes, of course, we have these CDC approved mosquito samplers where they emit carbon dioxide and attract mosquitoes. But leeches are only attracted by movement and body heat. So this year we were thinking of experimenting with those things. In fact, in the first stage, we wanted to experiment with those, but when Terence started his work, that was the period when we were at the peak of COVID pandemic. So I couldn't visit BSBCC, and we couldn't do much of these experimental things. But of course, we would love to have a trap for leeches, where we just go in the grid and do systematic sampling of leeches.
This is an exciting new thing. I remember when bear hair snaring first came out, as a similar kind of a talk somebody gave with, maybe we could collect hair from barbed wire hair snares. And you're at the cusp here of inventing something that's, potentially groundbreaking.
Yeah, thanks.
Hi, that was
Hi,
so
Ingrid.
nice to hear all your work since we spoke last time.
Thanks.
I just as this small briefing, mine was on flies, so it was a similar thing. I just went through and also looked at a little bit. It was a temperate region, so I was looking at brown bears. and even between, for example, the studies that have been done in the tropics on flies and the findings that we had here, there were differences in those species that we would get and how long they would have the DNA on them. But I think it's amazing to look at the leeches, especially if we can find a more systematic way of trapping them, because it's really difficult what we're looking for in the flies. It's more the meal rather than the blood meal. It's just the scat genetics that they are absorbing. So it's maybe a day in comparison to maybe 120 days. Some of my longer samples were maybe 2 and a bit days, but really, especially the ones in the field, it just degrades so fast, and I'm guessing in the tropics even faster potentially. So looking at leeches there would be amazing. Do you know if someone has done something to look at how far they travel as well after they fed?
As far as I know, they don't travel much. They don't travel as far as flies.
That's
So
really
again, we don't have empirical information because their travel is dependent on their host.
yeah.
And they on their own, they don't travel much.
This is really positive, especially for the
Exactly.
analysis that you do afterwards. And I played around a little bit with occupancy modeling and compared it to just scat surveys. And it is really accurate, even in the 1 kilometer grid square that I had. But I guess leeches are even more of a closed location. So that would be amazing to do. And you will get really accurate results of even individuals. So potentially you could even do behavioral stuff afterwards as well.
Probably, yes, because leeches, they just fall off at the place where when they are completely fed, and then they just, yeah, exactly, they just rest up.
Oh.
Yeah, so I think that's the most exciting part, because they are ideal samplers. They are moving blood banks, and if they can store the DNA, good quality DNA, up to 3 months, that means even if you're collecting leeches which are older and apparently very thin leeches, they still have those DNA. So I think, yeah, I think that's the most exciting part about leeches. But when you combine this with all these other samplers, dung beetles and flies and mosquitoes, I think that'll make the story even more interesting.
Sandeep, can you tell us a little bit about the detecting sunbear DNA in the claw mark? Because I think this is quite interesting, because often I get reports of people seeing bear sign, seeing claw marks on trees, and then we're pretty sure it's a bear, right? Whereas if you're just
Yeah.
going around picking up leeches, you've got lots of samples to analyze. Is that an easy sample to collect, or is it
Yeah.
complicated?
So sampling wise, it's fairly easy to collect. Unfortunately, we could not do trials with that. We just had, so whenever we got fresh marks, we went there and we did a swab sampling. So we took this cotton swabs and you dip that in ethanol and you move it up and down close to the chlorix, and then extract DNA, and you can identify the species. But we could not do, again, controlled trials with the aging. And if there is an opportunity, I think that'll be really good to do that kind of work. The rest of the method, by the way, was almost same. Only the sampling was different. And I think, to elaborate more on that, the idea actually came from our previous work on tigers in tropical Asia, not tropical Asia, subtropical Asia and central Indian Islands, India, where we were collecting, because their chlorix are quite big and very distinct. And you also get hair samples from there. So just an experiment, we did some swabbing there and we could detect tigers. And during the workshop in Taiwan, we discussed about that idea and we said, okay, let's add this and see if it works or not. So fortunately, it worked. I think there is a question.
Yes, I'm gonna tell you the question from my colleague, Pai, in the chat. You can answer that.
Okay, yeah, she's asking that whether the genotyping by sequencing could potentially differentiate two bear individuals. The leech might have fed on a female and a male bear on the same trail. That's a possibility. That's true. Although the possibility is less, because once the leech has latched on an individual, host individual, it would not fall off that easily. But yes, there's a possibility, and that's what we are doing in the second phase. That's where we are collecting leeches from these enclosures at BSBCC, where they release multiple bears, and we have blood samples of those bears, so we know the reference genotype. And if we get mixed genotypes, then we would be able to detect it. So we don't have the answer yet, but we have done the experiment to answer your question. And then the second part of the question was potential development of bear genotyping chip to facilitate the same SNPs comparison across range countries. Thanks for asking this question. I think SNPs are exciting. Genotyping chip is exciting. The main constraint for us in Global South is funding. I think we have capacity. We have technical capacity, but we don't have funding. So if people can assure funding, then these things can be done. And bear genotyping chip would be much easy, because we already have chip genotyping, SNP genotyping chip for brown bears. There's one more question by Miriam. The IUCN has worked a lot on biodiversity assessment with leeches. Jan Exner would be one to ask about leeches and how to trap them. And if he does not know, he will know who does. Yeah, thanks for that. In fact, I think Jan, he works at this institute in Berlin, and I'm based in Berlin. So I'm in fact thinking of talking to him about leech trapping. And thanks, you like the talk. Thank you so much.
I'll just add, Sandeep, it's actually a common thing here like I'm in Minnesota, there's a lot of fishermen here, and people often trap leeches. They just make their own little cans with holes in it, and they put bait in it. And this is how bait shops collect leeches. They collect hundreds or thousands of leeches. It's a big thing for people. That's the main fishing bait here.
Yeah. Thanks, Dave. Yeah, when I was looking for trap design for these leeches, I came across those videos about these leeches that they are trapping for fishing. But these are, by the way, these are aquatic leeches. And here in tropical Asia, we are talking about terrestrial leeches. So when I read more about that, they behave in completely different way. And for leeches, this is funny, but for leeches and mosquitoes, when you read about that, and this is, in fact, for mosquitoes, it's there in CDC manual also, they call us, they have this sampling method that they call on your body. So you wait and sample mosquitoes on your body. You wait and sample, leech sampling from your body is excellent, but then it has your blood. That you can basically, you can block it and still try and find out sun bear blood. But then I think it would, in my opinion, it would complicate the downstream procedures. So if we have a sampler, then that's the ideal situation. There's one more. Oh yeah, it's just a comment. Thanks, Cohen. Glad that you liked the talk. And yes, we will. As I said, we need funding for next phases. So thank you so much. We would definitely submit our research proposal. But I think I would also take this opportunity, since we have many people over here. It's a collaborative process. It started with this idea, this spark that was generated in Taiwan. And fortunately, we had Wong. He's not here today, but fortunately, we had Wong and Shamla there in Malaysia. So in fact, the workshop was in November, and I still remember the deadline for submission of this proposal, IBA RNC grant, was within 2 weeks. And we quickly worked on that, and we got the grant. But that was 2019, 2020 till 2022, we couldn't do much because of COVID pandemic. But now the pandemic is over. And if we have interest from the group, we would definitely like to replicate this at other sites. We can train people. Angeliki is here. We can train people and we can replicate that at other sites. Then it would become, it would go beyond the proof of concept study. Then it would become more useful.
For sure, I'd love to do this in Vietnam, where we have a sanctuary with Asiatic black bears and sun bears in the same sanctuary, and also at least one wild bear out there somewhere in the national park that I'd love to try and find.
Fantastic. Thanks, Brian. Yeah, so we'll talk more about that. And one more thing, I think, from this study that was done in China, they did the citizen science sampling. So they were actually, if you read that paper, they talk about involving rangers, although they were paying them, but they involved rangers, and if they get certain number of leeches, they get certain amount of money. But if we can convert that into a citizen science project, then we get more leeches from people, and that would be good.
Yeah, actually, I'm gonna say that. I mean, in at least in Thailand, the park ranger need to patrol, then you get the free sample from them
Yes,
somehow.
exactly. Yeah. And if I'm sure they're going for these samplings on systematic basis, like systematic time period. So it's kind of systematic sampling for capture recapture.
Yeah, even that, because one of the model is just like a random get the sample, and then you can analyze that in SCCR framework too. So
Yeah,
we not
yeah,
necessarily to be set up people in a systematic way.
yeah, yeah.
Any more questions for Sandeep or comments about the presentation?
Do you know, Sandeep, if the leech has to be attracted? You said it has to be a moving animal. Does it have to be like a warm moving animal, or will it be attracted to bait?
Yeah, no, that's another interesting question. So when you look at, again, disclaimer, I don't know much about leeches, but my experience in Asia and Southeast Asia, they're waiting for the prey and detecting the movement along with these thermal signatures, terrestrial leeches specifically. And as soon as you brush off the vegetation, they're on you. So that's the basic mechanism. But then just 2 weeks ago, this very interesting video came up where they found out, you can Google it, or maybe I can share the link later, but you can Google it, where they found the first evidence of leeches jumping. So they're actually recoiling like a snake, and then they're jumping from that leaf for the prey. So that's really interesting. That opens up this. So they're actively searching for the prey, and if they're doing it, that'll be really interesting for this kind of sampling.
But you could, for example, go into the forest with a dog and just have it running around in the brush for a while.
Yes.
Sacrifice the dog and then check it for leeches.
Yeah, that's true. I mean, yeah, we can try that.
Is there some idea of how many blood meals they would need as well? Because I guess the other danger is that you're getting a hungry leech rather than a leech that already fed on something. So you want them after they fed on something. I was just wondering if there's like a way where we could have, I don't know, stations that are attracted to bears, that bears are attracted to, but leeches just find them disgusting and they fall off. Then you just go and collect. And then it's like really low as well.
Yeah, ideally, yes. But the first part of your question, when you said that they are actually actively moving and searching for the prey when they're hungry, because when they're fed, they just drop off and they're under the leaf litter. And I had the same question when we started this work. But if you look at these papers that are published on mammal communities using universal markers for vertebrates, they actually go and search these leeches, and they're of different size and shapes. And then they make something that they call leech cocktail. So all the leeches collected from one grid are made cocktail, and then they do the analysis with that. So, and they find all these different species. So does not mean that the hungry leech that we presume as hungry leech does not have blood meal. And with that temporal experiment that we did from 0 day till 120 days. So on 0 day, when they're fully fed, they're as big as your thumb. But after 30 days, they're very thin, and then they don't change their shape or size. But then we could detect the DNA even up to 90 days. So Terence is unfortunately not here because he did all of those. He's the leech ringmaster. He did all of that work, and he made sure that those leeches survived. That was another challenge. But he took all the measurements. So we have, before we cull the leeches, we have their length, we have their girth size, we have their weight. We also did, again, I did not have time to present all of those things, and I was not sure whether it would be really interesting for you. But we have information about dung beetle size, dung beetle weight, and how it correlates with the DNA yield. We have the same information for leeches and how it correlates with the DNA yield, even for species, by the way. So there are 2 species of leeches that are often found in that part of Sabah, tiger leech and brown leeches. So we also divided that into 2 cohorts. So 4 bears were given brown leeches and 4 bears were given tiger leeches. And there were certain differences because previously published studies, they found out that there is prey-specific preference among these 2 species of leeches. But in our case, in control experiment, we didn't find much difference. Yeah, so I think there are several interesting parts of this leech work, and there are several unanswered questions. So again, I would call for collaboration if you guys want to collaborate, want to do some exciting leech work in tropics, you're welcome.
Thanks, Sandeep. Any more questions? Otherwise, we have a second agenda. Looks like not. So, Brian?
Thank you so much. Thanks, Brian and Dusit, for inviting me for this talk. It was really interesting. And I mean, it was really interesting to interact with all of you and present this work. So thank you so much.
Thank you so much for volunteering to be our first SunBear webinar.
Anytime. Thanks.
And for your patience. It took a while to organize as well. It took a couple of months to get into the calendar. So I'm glad it's finally happened.
In fact, I was waiting for Terence to finish his master's thesis. I didn't want to talk about his work before